Hello, my name is Melissa Latta. I'm the Principal Radiochemist at the Royal Brisbane Women's Hospital. Today I would like to share our experience in developing and producing a PET radiopharmaceutical for cholinergic imaging. By way of an introduction, I'd like to provide an overview of the facilities at the Royal Brisbane Women's Hospital Radiochemistry Laboratory. The main purpose of the laboratory is to supply high-quality radiopharmaceuticals for clinical use both in imaging and peptide receptor radionuclide therapy. The laboratory is a statewide service for PET radiopharmaceuticals and is TGA licensed for the manufacture of FDG and F18 as an API. We support both clinical services as well as research programs. A couple of years ago, the laboratory went through a significant upgrade where a new Grade C cleanroom was constructed, which is comprised of 8 hot cells, 2 dispensing cells, and is shielded by a safety cabinet. This cleanroom is complemented by a quality control laboratory, which contains all the contemporary analytical instrumentation expected to do quality control testing according to monograph standards. Further to this, we have a dedicated research and development area, which has a further 7 hot cells, 1 dispensing cell, and a full-size PET shielded by a safety cabinet, which really allows the facilitation of manual radiochemistry processes. We are also growing a second quality control area dedicated for research activities, and in this regard have recently acquired a HPLC and UPLC, where we expect this to be a full quality control laboratory this year. This slide summarises our current portfolio of radiopharmaceuticals produced in the radiochemistry laboratory at the Royal Brisbane Women's Hospital. Currently, there is a total of 16 processes validated at our site across our automated radiochemistry modules, and so I've split the radiopharmaceuticals up according to the module that they're manufactured on. So you can see on the left-hand side we have fluorinated radiopharmaceuticals made on our FastLab modules. In the middle column we also have fluorinated radiopharmaceuticals made on the Neptus module, and in the right-hand column we have our radiometals, both for imaging and radionuclide therapy, which are produced on the Syntomics module. The common theme to all of these processes is that they're all validated according to a kit-based approach to manufacture. And so what I've shown in the top left is a representative commercial kit which might contain a cassette and or consumables and or reagents to support a process. These kits are supplied or purchased from commercial vendors. They're designed to be single use and they're typically supplied with a validated sequence. This approach is not only convenient but also has advantages in process reproducibility. So if we now return to our list of radiopharmaceuticals in the current portfolio of the radiochemistry laboratory at the Royal Brisbane Women's Hospital, you will have an understanding of the concept of kit-based approach to manufacture. So each of the radiopharmaceuticals listed here has been validated at our site as well as subsequently produced using a sequence, that's a radio labeling sequence, that has been designed and developed by a commercial vendor and or supported with a kit which may comprise the cassette and or hardware or consumables and or reagents that are needed to support this process. The only exception to this is the fluorinated tracer FeOBV, which I've highlighted in yellow. You'll note that it's also in the Syntomics and the Neptus platform columns, and this reflects the original work being done on Syntomics, but more recently we've moved this across to the Neptus platform. So that brings me to the main part of our presentation, where we plan to share with you our first experience of developing a radiopharmaceutical when a commercial kit is not available. Several years ago, the laboratory was approached to supply FeOBV, a fluorinated radiopharmaceutical, for a clinical research program. FeOBV is a diagnostic PET tracer for measuring basal forebrain cholinergic function, particularly in Alzheimer's disease. More recently, it's also finding applications in imaging for Parkinson's disease. The original request was to support a study called Sterling Stream, which is led by Dr. Eamon Ellis. The study was proposed to be a 60 patient study where each patient would receive a 240 mbq dose of FeABV and then 90 minutes later a PET MR image. From the lab's perspective, there was no known monograph for FeABV. It was not available anywhere else in Australia, and as I've already mentioned, there was no known commercial kits across any platform, even ones that we don't have in our laboratory, available commercially. When the lab was presented with the challenge of implementing FeABV, the starting point was really to do a literature research and try and have a better understanding of what approaches have worked worldwide, both clinically and preclinically, to produce FeABV. Perhaps the most three relevant references that we found relevant to what we implemented at the Royal are listed on this slide. So the earlier report of Mulholland and others was a nucleophilic radiofluorination of a tosylated precursor to produce the product. This also required reverse phase HPLC purification as well as buffer exchange to formulate the product. The reported non-decay corrected yield was just under 10%, but the product purity was high, greater than 99%. Quality control testing was also reported in the literature, which is supportive of the standards that we produce all other fluorinated radiopharmaceuticals from. Our next challenge was to adapt what we had learned in the literature to make it essentially work or fit the equipment and infrastructure that we had at the hospital radiochemistry laboratory. So what we had learned was that we needed a module that was capable to write sequences. As I said, one wasn't commercially available for this product. So this meant a module that we were able to write all the steps to achieve the radiolabeling process. But we also needed a module that had HPLC purification capability. So a module that had integrative, semi-preparative HPLC purification so we could isolate the product pure enough to be used clinically. At the time of starting this project, the only module that was in use in our laboratory that fit both of these criteria was the Syntomics GRP synthesizer. More recently, we've acquired the Neptus Perform modules, and this platform is also able to meet both of these criteria. We also learned that the precursor required was a tosylate precursor called TEOBV, or in the ABX catalogue, TEBV. So this also alludes to that we were fortunate to find a reputable supplier for our laboratory, ABX, was not only able to supply the precursor required for this transformation, but they also provided a cold reference standard of the product so that we could use this in QC testing to determine the retention time they would expect for the product. As I mentioned earlier, all of our modules at the Royal Brisbane and Women's Hospital follow a kit-based approach to synthesis. And while a kit was not available for FeABV, we still needed a kit of some form to use with the module. They don't work without a kit, as opposed to some other research modules that are a little bit more traditional in scope. So in the early days, we essentially purchased kits for other F18 radiopharmaceuticals and modified these. So in the very first instance, we either bought F-MISO or FET kits, whatever was in stock with ABX, for the Syntomics module, and removed the components that we didn't need and added the parts that we did, whether that's consumables, hardware, or reagents. And we followed this same principle with the Neptus module at a later date. The other part of the process that needed to be developed on site was to actually write the sequence. Most fluorinated radiopharmaceuticals conceptually follow a similar process, and I've tried to label the key steps in the diagram at the bottom. So once you've generated your radionuclide, so in this case we're generating F18 from the cyclotron, we deliver that to the module. We then trap that radioactive fluorine on a QMA cartridge to recover the O18 water. We then remove, or elute I should say, the F18 from the QMA to the reactor and dry this through an azeotropic process. All of these steps are automated and are what we're writing to operate the valves in the module. Once we've dried the fluoride, we then add our precursor and do our radio labeling step. For FUBV, the next step was to transfer the reaction mixture for purification. As I said earlier, this required HPLC purification, which was something that we weren't routinely doing at the Royal at the time of starting this tracer. Once we'd been able to isolate the product, we also needed to do a buffer exchange because the mobile phase buffer was not suitable for product formulation and clinical use. So this required the product to be trapped on another solid phase extraction, eluted and formulated with the product formulation before sterile filtration for the final product formulation. I've tried to highlight schematically, at least with the symptomics diagram, how the module needed to be prepared in the way to meet the sequencing steps that we've developed. So this slide aims to summarise the process that we follow in our laboratory to implement a new product or a process. Up until now, I've been describing work which we would class as falling in the pre-validation workup. So this is the testing and process development that we undertake to define a reliable and reproducible process to make a product that will pass our quality control testing. Once we're confident we've got that process defined, we then move into the validation phase of the project. The first phase of this would be to write a validation protocol, where we identify all the key parameters of the process and conduct a risk assessment of the process. We also identify our sampling plan, and it's important to identify a sampling plan that's extensive enough to allow your product expiry to be determined. We also list all of our quality control testing and, very importantly, the corresponding predefined acceptance criteria. So to give an example of this relevant to FUBV, I've listed out our both release and post-release quality control testing below. You'll see the tests are very similar to the QC tests for most other radiopharmaceuticals in our laboratory. Typical things like product appearance, pH, radiochemical purity, sterility, all of the common things that are listed in, and the monograph limits are applied wherever possible. Or in the case of FUBV, information that were gathered also from the literature. Once the protocol has been reviewed, we then move into making or producing our validation batches. So the risk assessment will determine the number of batches appropriate, but for a new product, we're typically making no less than 3 consecutive batches that are made according to the clinical, or a scenario that represents a clinical production. So really using activity levels that represent what would be required to make patient doses. And that each of those batches then passes has a full quality control testing and passes all the criteria. The data is collated and written into a validation report, which is then reviewed by our quality assurance team. Working in a laboratory that follows GMP principles, all of our processes are documented from start to finish, and as such, implementing or revising a process will require new or revised documentation to be published as well. The final step prior to actually using or producing a patient dose is to write a change control, which is the final step to implement the new process. In the case of FUBV, I present here a table of data from the validation on the symptomics module. We did 3 consecutive batches, which are tabulated here. Through our pre-validation workup, we were able to determine that delivering less than 70 gigabecquerels of fluorine resulted in a more consistent process, and we're able to produce about 5% yield. This is a little bit lower than Mulholland's report of 9% in the literature, but we were producing enough activity to meet the requests for the clinical trial, or meet the requests for the patient doses for the clinical trial. All of our batches passed all quality control testing, and in fact all data was quite consistent amongst all 3 batches. We're also able to assign a product expiry of 7 hours from endosynthesis from HPLC testing of the bulk vial at this time point. Moving from validation to actually producing doses for a clinical research program, I've tabulated our summary of our data, and you'll see that the first batches were made on the symptomics module, the more later ones on the Neptus, but we were able to produce a high quality product which, while the yield has varied considerably, is sufficient to meet the patient doses requested. What I haven't included in this table is the batches that failed, so we're actually at a process reliability rate of about 70%. The batches that failed both on lower radiochemical purity in 2 cases, and in 3 cases either operator or module error. What I hope that I've shown you today is not only an example of how we've grown the radiopharmaceutical portfolio of our laboratory, but also our ability to support novel research by developing a process for a radiopharmaceutical that had no other supply chain. Specifically, that our site has developed, validated, and implemented a production process across 2 radiopharmaceutical platforms for the production of FeOBV used in clinical research. There is ongoing demand for FeOBV, and in this regard, we're always looking at continuous improvement of our processes. We'd like to increase our process reliability from 70% to above 95%, mostly for the benefit of patients not being inconvenienced by production fails. We'd also appreciate improving our product yield, even just a little bit more to approach the literature values. And we're also looking at how we can improve our process economy. And in this regard, we've very recently designed our own cassette and had it supplied by ABX in a gamma irradiated form for use on the Neptus platform. So this will just mean we're not wasting as many components being discarded from other commercial kits. And in ending, I just wanted to acknowledge the Department of Nuclear Medicine at the Royal Brisbane and Women's Hospital, where all of this laboratory work was done. HERF, the Hurston Imaging Research Facility, where all the PET MR imaging for the study was done. We acknowledge all the patients and contributors to the Sterling during the study, led by Dr. Ellies. Dr. Ellies presented the lab with quite a challenge when he requested FeOBV, and we value the collaboration with our laboratory for this project. Finally, I've also included a snapshot of this very recent publication if anyone would like to read more about this project.